Okay, so this video is going to be short and sweet, I promise. One thing that we looked at, and we looked at this when we talked about proteins, there are certain organelles in the cell that actually have to do with protein synthesis. So we give these a specific name, and we talk about how they all work together to actually enable proteins to be made and to transport these proteins throughout the cell. So let's take a look at what they do. All right, so some of the parts, your nuclear envelope, of course, we know that's going to play a part because remember, our nuclear envelope is the reason why DNA cannot leave the cell. So DNA has to make mRNA, which leaves the cell. So that's very important in protein synthesis. All right, chromatin, of course, these are your chromosomes, and this is your DNA. So we need those. Your nucleolus is important because some RNA is made there. We'll talk about that. Your nuclear pore, these are the holes in which the mRNA leaves. So we're going to need those as well. All right, your ER. Remember, you have a smooth and a rough ER. Your rough ER has ribosomes, and these ribosomes are the first place that we're actually going to see those proteins that are made through protein synthesis. All right, so these organelles together are called the endomembrane system. So, what are the organelles involved in protein synthesis, and what role does each organelle play in protein synthesis, and what impact does it have on the process? That's what we're going to be talking about in this video. All right, so let's look at the organelles involved, and then we'll talk about what they do as we go. So here's our endomembrane system. You can see all of these are connected. And we've talked about the fact that you always find the rough ER close to the nucleus. Well, there's a reason for that. Whenever that mRNA is made, we need it to get to these ribosomes. So with it being right here, that makes it a little bit simpler. All right, whenever we see the word endo, that means within or on the inside. All right, the word membrane is a lipid bilayer. We talked about the properties of lipids and how they actually make the lipid bilayer. All right, so this entire system, its job is to regulate protein synthesis and to make sure we get the proteins where they need to go. All right, after the protein is produced, which we know the proteins are produced at the ribosome and the polypeptides are, and then they're folded, they're going to be used inside the cell, but some of them we need to get to the outside of the cell, and that's going to make a difference in where they go. All right, so there's a lot of different organelles that's involved in this process. We have the nucleus, we have the rough ER, we have ribosomes, we have transport vesicles, we have our Golgi apparatus, and we also have our cell membrane. So all of these are part of the endomembrane system, and they each have a role in protein synthesis, but they all have to work together to make this process happen. All right, so our nucleus, of course, we know this is where our mRNA is made. This is where transcription happens when we've already talked about that part and what actually happens during that process. So this happens in the nucleus. So, of course, we need the nucleus, and this is where our process is going to start. So this is where we find our DNA. DNA cannot leave the nucleus, so we're going to need that RNA. All right, now here, of course, we know that we're not going to have proteins because the proteins are made on the ribosomes, but the message for the protein has to come from the nucleus. So there's no proteins in the nucleus because there's no ribosomes in the nucleus. All right, so our rough ER is where we have all of these ribosomes, and that's what makes it rough. So after mRNA is made, mRNA leaves the nucleus through the nuclear pores, and it attaches to a ribosome, which is on the rough ER. So that's important because that's the first step after that. When that protein is made, we're making these proteins on the ribosome, which are attached to the rough ER. So not the free-floating ribosomes that are in the cytoplasm, but this mRNA is going straight to the rough ER in those ribosomes. All right, now, both of them, because they're both ribosomes, they can both make proteins, but what is done with those proteins is what's going to make the difference. So the ribosomes that are attached to the rough ER, the proteins that are made here, these are going to be used inside the cell, or they're actually going to be these free-floating ribosomes, that proteins that they make, so the ones out here, the ones that are made, they will stay in the cytoplasm, whereas the ones that are made here, these are going to go to other parts of your body or other parts of the cell. So they're actually going to leave. So they're made on the rough ER, they're going to leave the cell and be transported out. Whereas if the ones you have that are just found in the cytoplasm and not attached to the ER, those are going to make proteins that are going to be used within the cell. All right, so these are going to make proteins that are going to leave the cell. All right, so next, after we make our protein, our mRNA has gotten to the ribosome. We made our protein in the rough ER, because remember, the proteins that are made on the rough ER ribosomes, these are the ones that are going to leave the cell to help with other parts of your body. All right, so translation occurs at the ribosome and the protein is produced. So this is the first time we're going to see a protein, and that's in the rough ER. All right, so now after my mRNA is finished, after it has coded for the protein that it's going to make, it's going to be broken apart. And of course, we know it's going to be broken apart into nucleotides. So when we talk about DNA polymerase, how it takes nucleotides and it builds a new strand of DNA, or how RNA polymerase builds a strand of RNA, these nucleotides are just constantly recycled. So we made the mRNA, but then the mRNA will break down. And so now you have all these nucleotides just floating free in your cytoplasm. All right, so the next step. All right, so there's our little protein. All right, so now remember, the proteins that are made on the rough ER are going to be transported out of the cell. All right, so in order to do that, we're going to need a transport vesicle. So the protein travels through the rough ER and is carried in a transport vesicle until it gets to the Golgi apparatus. So remember, the Golgi apparatus is like the post office. It's going to package and it's going to prepare those proteins for transport. So it's going to be carried to the Golgi. And there is going to be packaged, it's going to be modified, and it's going to be sorted so that it knows where to send it, just like the post office. We sort packages according to where they need to go. All right, so the Golgi apparatus is like the post office. It packages, modifies, and sorts the proteins. All right, so now that protein will be sent to its final destination, just like our packages at the post office. They get to be sent to where they need to go. And that's going to happen in the Golgi. All right, so then that little transport vesicle, we talked about exocytosis, and this is what's going to happen to that protein. It's going to go through exocytosis to be put out of the cell so it can go to other parts of your body and be used. So a transport vesicle from the Golgi will carry that protein to the cell membrane where exocytosis will happen, and now that protein is on the outside of your cell and is ready to be used by your body. All right, so then it goes to the body to reach its target cell. All right, now if you remember during cell communication, we talked about the difference between protein ligands and lipid ligands. So this would be a protein that's about to go to its target cell. Remember, its receptors are on the surface of the cell, so that's going to be important. And y'all, that's it. Just like that. Those are the major players in the cell that have to do with protein synthesis. So I thought it was real important that you get this. Make sure you check for the right page in your notes for this. We've already filled out these notes, but just make sure that you move them over so that you remember when we talked about proteins, you remember that now this is where that process is happening and this is why it's happening. So I will see you in the next video. Have a great day.